Biodiversity and Physiology of Body Systems task booklet, and we're looking at nutrition. Question 9. So let's have a look at the diagram, and it tells you it's a gut. So this is heterotrophic, holozoic, and we've got food. This would be the kind of mouth end, and then we've got the anus end, and we've got 4 processes there. Number 1 is taking the food into the gut, so that is going to be ingestion. Number 2, we've got enzymes, and the diagram is showing us something big that's been broken down into smaller things, so that's digestion. Then the smaller things are then being taken in to the body of this animal from the gut. So that's things being absorbed, so absorption. And then the water's removed, and then what's left is then pushed out of the tube back into the environment, and that's egestion. If you get all 4 marks, all 4 correct, you'll get 2. If you get 3 of them correct, you're down to 1 mark, and if you only get 2, then you've blown it for that. Define process number 2. So define digestion. So this is just tell us what it means, and you need to say some key things in there. So on the diagram, we've got large thing going to smaller things, so that'll be part of the answer. But also the properties of this large thing against the small things, and what is it about the small things that allows the next process to occur. So we're going to breakdown of large insoluble molecules into these smaller soluble ones, and when they're soluble they can then be absorbed. So breakdown of large insoluble molecules into small soluble molecules. So just a definition in your notes. And now we extend this simple tube, and it tells us we are now in mammals. So, and you should now be thinking of the human gut and the different areas, and lots of enzymes and places in there. Okay. So hopefully you've learned these. You've made a nice table of enzymes and what they work on in your notes. So this should be pretty straightforward, pretty quick. Move on to another question that you're going to spend a bit more time thinking about. Okay. So this is talking about the stomach, and you could go into detail and say gastric pits, that sort of thing, but we'll stick with stomach for the moment. And pepsin is going to break down proteins, but it's giving you a clue here. Peptides, which are kind of smaller versions of proteins and polypeptides, so they just like not quite amino acids, but small chains. So we'll have protein there, or indeed polypeptide. And now we're taking these peptides and we're breaking them into smaller things. Well, what's the smallest sort of thing you can get there? The monomer is the amino acid. Plural. And where does that happen? Well, it's a little bit ambiguous here. Where does it actually get secreted? Well, it's the pancreas that secretes the enzyme, then it travels in a duct and then goes into the small intestine. So they'll accept either answer there. So pancreas, actually where it gets secreted, and then then it does go into the small intestine or the duodenum. So small intestine, there's a possibility there. And then we've got lipids and the products of that digestion. And again, just where does this occur and what's the name? So the name of the enzyme that breaks down lipids, we've got lipase. And then where does this occur? Well, lipase, again, it's in the pancreas, where it's secreted. And it will go into the small intestine. And lastly, hopefully you're really familiar with this one now, what produces maltose as a product. Well, we have got starch would go to maltose. So starch is a polymer, maltose is a disaccharide, so polysaccharide, disaccharide. And it's telling us that's in the salivary gland. So that all ties in, and the enzyme, you should be fine with this, amylase. Okay, instructions here in the spaces provided below. Draw and label 2 cells. So a common problem here is drawing far too many cells, wasting time. There's 2 marks for drawing and labelling. So we're looking for one of these cells. So some people have drawn this entire villus, but we need one of these kind of normal epithelial cells, and then very difficult to see on here, but there's some different cells as well, which are the goblet cells. So we are having the cell, and this can be just a simple kind of diagram. So this is our epithelial cell, and if we were putting something here, we'd label this as our microvilli, or sometimes called brush border, and we could put a nucleus in there. You could put mitochondria. That's the sort of thing that we are expecting there. The actual marks, and one for the diagram showing this brush border, and we'll do the function down here as well. So the correct function of this is for absorption in a large surface area. So absorption of digested products, something like that. Then the other type of cell we're looking for, not quite the brush border, but it's the goblet cell. So the one that looks like anything that looks a little bit like that, and we're going to call that a goblet cell. And the function there, to produce mucus. So this is the goblet, and function produces mucus, and that will lubricate the food and so on. Describe an adaptation found in one of the cells you have drawn that enables it to carry out its function. So if we were talking about this epithelial cell, then the most obvious thing to talk about would be this large surface area of the microvilli. So on the epithelial cell, we have microvilli, which gives us a large surface area. And it says describe an adaptation, so all we're doing is describing what we see and telling what the feature really gives us. Not a really detailed explanation, it's only for 1 mark. Other things we could talk about mitochondria for active transport. We could have, if we were talking about this goblet cell, so I'll put in the goblet cell there. So we could have that the goblet itself stores mucus. Stores mucus, and that would be to lubricate food, but we don't need to go that far. Okay, and that's 12 marks.